could you start by telling us about yourself, and that's the usual way we warm up things?
Right. My name's Anne Ferguson. I'm a retired consultant anaesthetist who trained a very long time ago. I spent a year of my training in Montreal working on reversal of anaesthetic agents, such as curare. I was a consultant in East Kent, and since retirement, I've studied the history of curare. I've given talks on curare so that I'm almost bored with the subject. And I have, I was a judge on the Golden Dagger for several years. I found retirement very interesting. Okay.
Okay, can you put your screen up now, please, Anne? All right, and can we go to the beginning?
There we are. There we are. Hang on.
So this is our talk, our much delayed talk, Ordeal being both therapy and poison.
Yes.
Over to you, Anne, and let's hope it goes well.
Thank you very much. Thank you for asking me to give this talk, and congratulations to Peter on managing to get as many people into this meeting as possible. It really is a difficult job, and he's done a grand job. Thank you, Peter. I have to declare before I start that, let me find, that's it, that I have given part of this talk before to the History of Anesthesia Society, and it was published in the proceedings in 2017, and I was due to give some of it this year, but I was ill, so it'll be given next year and published. But a lot of this talk is new for you. I have the editor's permission from the proceedings to give this talk. By the early 19th century, there was a great trade up the west coast of Africa in palm oil, ebony, and gold. And in Calabar, marked here in red, the Calabar bean was used as an ordeal poison, known locally as ezere or chopnut. And this was well established. Locals suspected of committing a crime or witchcraft were made to eat a bean, the effects of which were either very unpleasant or fatal. What would happen is their muscles would twitch all over and then paralyze, and they would salivate. This was called soap coming out of the mouth. The Reverend Hope Waddell followed this trade route, got some of these beans and took a story of them back to Edinburgh to Sir Robert Christison, who was Professor of Materia Medica and a very well-known toxicologist in Edinburgh. He examined the beans and the effects of them, and he wrote a paper in 1855. Rabbits were subjected to the bean extract and developed weakness of the muscles, muscle twitching, and feeble heart action, and then they died. But he thought the sensation was maintained. He couldn't work out why they died, so of course he tried some on himself. One evening he took 1/8 of a bean and retired to bed with a pleasant feeling of slight numbness in the limbs, like that which precedes the sleep caused by opium. That makes me wonder what else he was trying. The next morning, as he hadn't felt very many symptoms, he did a proper job and took a great deal more. And he experienced giddiness, so he went and had a shower. And after he'd had his shower, he realized that he was actually in big trouble and he needed an emetic. Now he was in his bathroom and the only thing available was this soapy shaving water, which he drank to good effect. He retired to bed and was very cold. Another toxicologist was called, but he couldn't do much apart from observe symptoms. He was cold, so they put a mustard plaster over the abdomen. His pulse became irregular, and he said that he thought that he was in a condition like that induced by profuse bleeding after delivery. What he meant was that he had a very low blood pressure, but of course at this time they couldn't measure it. He slept all day, then had coffee in the evening and felt much better. His conclusions were that the fatal action is on the heart, but there was an additional action of paralysis of the voluntary muscles, but that the integrity of mental faculties was most remarkable, and coffee was curative. And he thought that the possible uses were therapeutic, and it might be of use for humane execution. Please hang on to that. The beans were passed to John Hutton Balfour, known as Old Woody Fiber, who was a professor of botany. And in fact, medical students right up till the middle of the 20th century in Scotland had to study botany. Medicine at this stage was very plant-based, and botanists were very interested in any plant that might have therapeutic properties. So it was important to classify this plant, which Balfour did. He grew the beans, but they wouldn't flower. And so he got flowers from Calabar in spirit. And the most interesting feature of this plant was, this is the stigma, and there was a very unusual sac at the end of the stigma. Sorry. So he named the plant Physostigma. And the active ingredient became known as physostigmine. The beans were passed to Thomas Fraser, assistant to Christison, who in his gold medal winning essay of 1863 described the actions in great detail. He did a very large number of experiments, and he found, that's Thomas Fraser, and he thought that the physostigmine acted on the spinal cord. So he tried some beans himself, of course, they seem to all do that, on a very large number of animals, and made very detailed conclusions. But the thing that he did notice was that the pupils constricted. And this was immediately of therapeutic use. And he passed this observation on to Douglas Murray Cooper Lamb Argyle Robertson, an ophthalmologist. Now, what ophthalmologists were doing at this time was putting atropine in the eye so that the pupil dilated and they could look into the back of the eye. But they had no means of contracting the pupil, and atropine could last 5 days. So this constriction of the pupil by physostigmine was of a great advance to ophthalmologists. And Argyle Robertson said, there can be little doubt that in the Calabar bean, we possess an agent that will soon rank as one of the most valuable in the ophthalmic pharmacopoeia. The pharmacologists and physiologists got hold of the bean, and in 1864, Rudolf Lenz tried slowing the heart using physostigmine and found that the heart still slowed even if you cut the vagus nerve. So he concluded that it worked on the last part of the vagus nerve. By now, a large number of beans were being imported through Liverpool, and in August 1864 at Queen's Dock, the bark Commodore had unloaded its cargo of palm oil, Calabar beans and ebony and left a tremendous mess on the dock. This was cleared up and moved into a hole nearby, and these beans were found by 46 local hungry children and one pregnant adult, and were eaten by them. They were all admitted to hospital, and all of them showed the same symptoms that Christison had done: weakness of the muscles, increased peristalsis in the gut, and low blood pressure. They were given a zinc emetic, and if that didn't work, they were then given a mustard emetic, wrapped in warm flannel and nursed on their mother's knees. A very detailed report of this was written by the houseman, and I've had access to that, so I know exactly what happened to each child. One child died. The coroner said this must never happen again, and it didn't, until 1871, when rubbish in Coburg Dock from a similar ship was dumped in Boundary Street, and 17 children were poisoned. Emetics were administered, and all survived. I was very upset to read in an unreferenced article in the New Scientist that the reason that these children had survived was because they were all given atropine. I can find no evidence at all for this. So I wrote to the New Scientist and said, please could they let me know of all the references to this? I got no reply. Plants were important therapeutically at this time, as I've said, and attempts were made to use physostigmine for everything in sight. The only time it seemed to work was in some cases of muscle weakness. And I think that these were myasthenia because 100 years later, myasthenia was treated with physostigmine. So the records repeatedly show that these beans are toxic and that there are two systems involved. The voluntary muscles, that is those that we control to walk and to stand up and to talk, and the involuntary ones, which adjust the internal milieu. And it seems that these beans were affecting both systems. We need to know what physiologists were up to at this time, because apart from the use in ophthalmology, physostigmine was very much a physiological tool. Now, at this point in the talk, I tried to write it so that I was following the involuntary system to start with and then the voluntary. It was just a mess. So I'm sorry, I have to jump from one to the other. Oswald Schmiedeberg, working in Strasbourg, tested the effects of the bean and compared them with the toxic effects of Amanita muscaria, which you may have seen as you go walking. And the effect of these beans was henceforward known as muscarinic because it reproduces the same effects, and the effect is inhibited by atropine. In 1867, chemists got interested, and Jobst and Hess showed that physostigmine was an alkaloid. Well, search me, I'm not a chemist. In 1870, Fraser, who we have met previously, published a paper on atropia as a physiological antidote to the poisonous action of physostigmine. It seems that they tried to dress the names of these drugs up the whole time. He gave a series of rabbits a fatal dose of physostigmine plus atropine, and they all survived to live another day. Until a few days later, he gave them the same dose of physostigmine with no atropine, and they all died. So from which we can conclude that the atropine blocks the effect of physostigmine. Now, the voluntary system was investigated by Claude Bernard in Paris in 1850. And he was working on frogs. Now, Professor Dronsfield says I get too excited about frogs and I show too many. So I'm only showing one today. What Bernard did was to isolate the sciatic nerve on a frog and then tie the limb beneath the nerve so that the blood vessels to that limb were occluded. He then stimulated all four limbs of the frog and it moved all four limbs. He then put curare into the incision on the frog's back to paralyze it. And after a few minutes, it only moved the ligated limb. And his conclusion was that curare only affects the motor nerves and only at the nerve endings. Powell, working 50 years later, was working on animals that he'd anesthetized and paralyzed with curare, and was then experimenting with physostigmine on the gut. But as soon as he gave the physostigmine, the curare wore off, from which he concluded that physostigmine antagonizes curare. In the involuntary nervous system, there is a steady state which can be tipped either way. There's the sympathetic system, there's the fight and flight reaction, putting our pulse up, and I'm in this state at the moment. My blood pressure is up and I'm sort of slightly anxious. The other is the parasympathetic, the rest and digest. And I hope that those of you that have got into this meeting this afternoon are in the rest and digest system. You're sitting comfortably, your blood pressure's low, your pulse is down. The fight and flight prepares the body for action. You can makes you want to go into battle. It increases the pulse and blood pressure, stops the gut. Well, it'd be very inconvenient if you were dressed like this and about to charge the enemy and you needed to get off and go behind a bush. So it's as well it does stop the gut. This system doesn't enter our argument anymore. The rest and digest, as I've said, prepares the body for rest and digest. And this is the system that we are looking at as regards the involuntary system. And these are the muscarinic effects. John Newport Langley was a physiologist and spent 50 years in Cambridge as a physiologist. He was a very careful man and didn't come to conclusions easily. But he was very interested in this involuntary nervous system, and he spent 30 years working on it, and working on it anatomically, pharmacologically, and physiologically. And then he called it the autonomic nervous system, and it's still called this today. He then worked on the effect of poisons on them. I must emphasize at this point that there are a lot of poisons that act in exactly the same way as physostigmine. One of these is jaborandi, which comes from Brazil. Now, you should all be growing this and you should sprinkle it round your doorway because it keeps evil and no good neighbours away from your property. What Langley did was he examined the effect of atropine on the heart rate and put it up, which one would by now expect, and jaborandi, which contains pilocarpine, which also constricts the pupil, put the rate down. On dog salivation, pilocarpine increased the salivation, atropine decreased it. And he concluded that the end effect depends on the relative concentration of the two drugs. And he described this as his mutual antagonism of poisons. And he wrote, "We may, I think, without much rashness, assume that there is a substance or substances in the nerve endings or gland cells with which both atropine and pilocarpine are capable of forming compounds." This was the start of his receptor theory. Now, he thought that a nerve came to a cell, it went into the cell. He was wrong about that, but he was right about the rest. It acted on a receptor substance in some way, which then made the chief substance of the cell do whatever the cell did, like muscle contract or secrete or whatever. Paul Ehrlich, working in Germany, came to the same conclusion independently in 1878, although it was only Langley who thought that drugs might act on these sites. And Ehrlich formed the word chemoreceptors. Langley then started to work on the voluntary muscles. And he found that whether he used intact nerve or denervated nerve, you give nicotine and this contracts the muscle, give curare, the contraction's blocked, but galvanize the muscle directly and it still contracts. So both drugs act somewhere on the muscle, but not on the nerve, but not on the bit that contracts. He said, since neither curare nor nicotine, even in large doses, prevents direct stimulation of muscle from causing contraction, it is obvious that the muscle substance which combines with nicotine or curare is not identical with the substance which contracts. Langley's pupil, Elliot, speculated that the nerve endings on striated or voluntary muscles were different to those in the autonomic nervous system, and he coined the term myoneural junction, which we still use today. And he was the first to suggest chemical transmission between a nerve and the end organ. And Langley said, in my view, the myoneural junction is part of the receptive substance localized on the neighborhood of the nerve endings. He presented his findings in 2007 in Heidelberg and came up against Rudolf Magus, who had repeated Langley's experiments and come to different conclusions because instead of nicotine, he'd used physostigmine because that stimulated muscle before it made it paralyzed. But of course, we know that Pal had shown that physostigmine antagonizes curare. So it's not surprising that he got different results. This stimulated the soup or sparks debate. In the parasympathetic or the voluntary muscles, was the transmitter chemical, soup, or electrical, sparks? Nobody really knew. Years before, Émile Dubon-Poiremont had suggested that it was a chemical, and Ernst Bruch had said transmission by chemical mediators was like a lady with whom the neurophysiologist was willing to live and to consort with in private, but with whom he was reluctant to be seen in public. Most physiologists thought that the idea of chemical transmission was quite unthinkable. Dixon, in 1907, did this experiment. He used extract of a dog's heart that was inhibited by vagal stimulation and put it into an amphibian heart. And you can see that the heart slowed and that this was reversed by atropine. Unfortunately, he didn't write this up. And Dale wrote later, it does seem to indicate as early as 1907 that chemical transmission was a possibility. Dixon gave this lantern slide to Dale before he died, and Dale published it saying that it was Dixon's work. The research for and belief in chemoreceptors went very quiet. And I think this was partly due to these clashing results and partly due to a lot of these physiologists going on to be trained as doctors, which happened to Dixon. A world war intervened and a lot of them enlisted in the services. Dixon went into the Navy and spent his entire naval career of the duration of the war in that rather non-maritime city of Madrid. It was neutral, and a lot of agents from both sides were there. The word spy has been used. Nobody really knows what he was up to there, but he got an OBE for it. So there was still no explanation of why stimulation of the vagus nerve, part of the parasympathetic system, as I've said, caused these effects, which I've already described as muscarinic. We now need to look at acetylcholine. Acetylcholine was first synthesized by chemists in 1867, but was regarded as merely a synthetic compound. Reed Hunt tried it on a body in 1906 and found that there was a profound fall in blood pressure, which was very fleeting. Dale had worked in Langley's lab, and he moved to the Wellcome to work for commercial reasons on ergot of rye. He found in it a very rare contaminant. It depressed a cat's heartbeat, just like muscarine, and he wondered whether this contaminant was acetylcholine. And this was shown to be the case by a chemist, Arthur Ewins, in 1914. Dale wrote at the time, acetylcholine occurs occasionally in ergot, but its instability renders it improbable that its occurrence has any therapeutic significance. And in 1914, Dale showed that acetylcholine had both nicotinic and muscarinic effects rather like physostigmine. He found the effect of acetylcholine very fleeting, though, probably due to hydrolysis, and also found that it could be blocked by atropine. Otto Loewe, in 1921, did a landmark experiment. This was very important. He took 2 mammalian amphibian hearts, isolated. One had the vagus attached, the other didn't. And he perfused the first one with ringolactate using stimulus to the vagus. He collected the perfusate, and in discontinuity, so there could be no transmission electrically, put it through the second heart, and it contracted. And he found that atropine blocked this effect, and ezerine, which is another word for physostigmine, they're exactly the same thing, intensified it. He had the greatest difficulty in persuading his colleagues of a totally new concept, that physiological action of an alkaloid could be explained by inhibition of an enzyme. His discoveries were thoroughly exploited and found to hold for the whole parasympathetic system. But it still wasn't realized that acetylcholine was a naturally occurring substance until Dale found it in a horse spleen in 1929. And he wrote, "It appears to us that the case for acetylcholine as a physiological agent is now materially strengthened by the fact that we have been able to isolate it from an animal organ, and thus to show that it is a natural constituent of the body. We have yet no conception of the meaning of its presence in the spleens of these large ungulates. And as far as I know, we still don't know." Mathis, working in Dale's lab, showed that acetylcholine in blood works for a microsecond, and then it's destroyed by an esterase. And Feldberg, working in Berlin, he was Jewish and he was in Berlin. He moved permanently to London, and he brought with him the knowledge of physostigmine. And eventually Dale wrote, "We thus came to regard physostigmine, or ezerine, as an indicator for the action of an unstable choline ester like acetylcholine." Dale wrote that the nicotinic action of acetylcholine was only just starting to be investigated, and Adrian put his head on the block by saying that he thought it was the same process at neuromuscular end plates. In 1936, Dale showed that stimulation of motor nerve fibers to perfuse voluntary muscle causes the appearance of acetylcholine in the venous fluid. And when transmission of excitation from the nerve to the perfused muscle is prevented by curarine, stimulation of the motor nerve fibers still causes a release of acetylcholine. And that if he didn't curarize it, but just stimulated it for a bit, acetylcholine would appear. And if he then stopped stimulating, the acetylcholine would disappear and then reappear when he started to stimulate it again. So we've now got the system that stimulation of a nerve, whether it's vagus to the heart and gut or a motor nerve, causes release of acetylcholine at the nerve endpoint. the acetylcholine is destroyed by cholinesterase and the whole system starts all over again. If you give physostigmine, that inhibits the cholinesterase, so the acetylcholine is no longer destroyed, so it continues to act. And you get an effect of an overdose of acetylcholine. The muscles will twitch and eventually fatigue, and the heart will go very slowly. Physostigmine was still being imported. It was not yet synthesized, and it's very unstable. And in 1931, a group of chemists at Roche in Switzerland decided to find a more stable replacement. They eventually synthesized neostigmine. It's more stable, it's synthesizable, but it still has to be kept in dark brown ampoules. At that time, it was still a physiological tool, and I don't even pretend to understand this paper. So why were anaesthetists interested in all this science? Well, this is a nerve from this is a motor nerve. You can see at the end of the motor nerve the acetylcholine vesicles. Stimulation of the nerve causes the vesicles to be released, and they go onto the acetylcholine receptors on the muscle end plate, and this causes the muscle to contract. And it's these acetylcholine receptors that are blocked by curare. At about this time, it became easier for anaesthetists to be able to put a tube down a patient's throat so that we could ventilate them into the trachea, that is, to ventilate them. And we were developing the means to be able to keep a paralyzed patient alive easily. So anaesthetists started to do this, to paralyze the patient. This had enormous consequences. The patients did better because surgeons could get further into the patient's body much more easily. The patients didn't have to be given nearly so much ether or chloroform or whatever, and so they woke up more quickly. It really was a revolution in anesthesia and indeed in surgery. So, at the beginning of an operation, not all operations, but some where it's considered appropriate to paralyze the patient is releasing acetylcholine into their muscles as usual, even if they're anesthetized. Give curare, it blocks the action of acetylcholine, so the patient is paralyzed for as long as possible. And I've done this for over 12 hours for an exceedingly tedious operation on several occasions. You have to keep topping up the curare, but you can do it. At the end of the operation, neostigmine is administered. It increases the life of the acetylcholine. Acetylcholine increases at the end plate, and nerve function returns. Physostigmine would have done just as well as neostigmine, except that it was no longer available. But there's a snag. Neostigmine decreases destruction of acetylcholine at both muscarinic and nicotinic sites. And at the nicotinic sites, this is the effect you want, and it reverses the curare. At the muscarinic sites, it causes a bradycardia, so the patient has to be given atropine as well. And I did this throughout my clinical career. We replaced atropine with glycopyrrolate, but it was the same system the whole way through. I'm now pleased to say that chemists are developing more specific drugs, but I'm not the right person to talk to you about those. One other interesting aspect of acetylcholine is that if you stick two molecules together, end to end, you develop a drug called succinylcholine. And this, administered to an anesthetized patient, they'll twitch and then they'll paralyze for a few minutes. And you can get a tube into their trachea, or you can give them ECT, or reduce a fracture, or whatever. This succinylcholine is then destroyed by the patient's cholinesterase, except that sometimes it isn't, and you find you've got a paralyzed patient for 45 minutes, which can be embarrassing, but that's life. But as I say, chemists, thank you very much, have synthesized much newer and more effective drugs. So let's return to Christison and his Calabar bean. I'm ever so sorry to have to remind you that in 2001, the headless, limbless torso of a child was found floating in the Thames near Tower Bridge. He came from the Benin area of Nigeria. I can only hope, and on post-mortem, he had Calabar beans in his gut. I can really only hope that Christison was correct when he said that Calabar beans might have a place in judicial execution, but I'm not very hopeful. I think that child had a rotten death. Thank you very much.
Thank you very much, Anne, for that wonderful talk. And again, I apologize to everybody for the problems we've had. Actually, as it happened, we haven't had any questions to that show what a wonderful talk it was, but everybody understood what you were saying. And that's really about it, I think. Again, I really do have to apologize for probably sending out the wrong link. And we hope that you enjoyed it nonetheless. And thank you, Anne, for being so patient and calm and eventually going ahead and giving such a wonderful talk.